What if we could program living things, just like we program computers? I'm not talking about Python or Java. I'm talking about writing code in the language of DNA itself. Now, that might sound like something straight out of science fiction, but it's really happening right now. We are on the edge of an incredible revolution, and today, we're going to open up the toolkit that's making it all happen.
And that's really the heart of what we're talking about today. See, this isn't just about understanding biology anymore, it's about actively engineering it. We are, for the first time, learning to read, write, and even rewrite the fundamental code that makes life, well, life. So, to really get a handle on this, let's stick with that computer analogy. If we want to program life, we first need to understand our toolkit, the biological hardware and software that we're actually going to be working with.
First up, let's look at the big picture. Biotechnology. This is just the super broad field of using living things for industrial stuff. You can think of it as putting biology to work for us. Everything from using yeast to brew beer, all the way to creating life-saving medicines. People have been doing this for centuries, really, but the tools we have today have made it unbelievably powerful.
Okay, now here's where the real programming part comes into play. Genetic engineering is the super precise tool inside that bigger biotech toolkit. This is how we can directly edit an organism's DNA. It's literally how we write our new biological code.
So if we're programming, what's our computer? Well, for a lot of biotech, the perfect hardware is actually a humble little microbe, like bacteria or yeast. And why them? Well, for one, they reproduce like crazy, which means we can go from a single cell to a giant factory-sized batch super fast. Two, they are these amazing little chemical factories, already great at making the complex molecules we want. And here's the crucial part, the interface. They have these little loops of DNA called plasmids, which act just like a USB drive. It's the perfect port for us to plug in our new genetic code.
Now let's talk about the software. Instead of thinking about a long line of code, it's actually way more helpful to think of a gene as a part of a circuit. Just like an electrical circuit has switches that turn a light bulb on and off, a genetic circuit has these tiny molecular switches that can tell a gene's function when to start and when to stop. And this genetic circuit is built from three really key parts. First, you've got the promoter, which is basically the on switch. It tells the cell, hey, start reading the code here. Then you have the coding sequence. That's the actual program, the instructions for making whatever protein you want. And finally, you have the terminator, which, you guessed it, is the off switch. It tells the cell, okay, you're done, stop here. It's this simple, elegant control system that makes all of this biological programming possible.
Okay, so we've got our hardware and we've got our software. But how do we actually put them together to create something useful? Let's walk through the actual process of installing our genetic program and then, running it at a massive scale.
The workflow is actually a pretty neat four-step process. First, you find and isolate the specific gene you want. That's your program. Second, you take that plasmid, our little biological USB drive, and you use special enzymes to snip it open. Third, you paste your new gene right into that opening. And fourth, you pop that newly modified plasmid back into the bacteria. And just like that, you've created a genetically modified organism, or GMO.
But having just one single program bacterium isn't going to do much for us. To actually make a product, we need billions and billions of them all working together. And that's the next big step, scaling up. We take our little modified microbes from the lab petri dish and we grow them in these huge industrial tanks called bioreactors or fermenters.
At that industrial scale, the process looks something like this. First, you get the bioreactor ready with all the food and nutrients the microbes need to grow. Then, you sterilize absolutely everything to make sure no other rogue bacteria sneak in. After that, you inoculate the tank. That's just a fancy word for adding your newly programmed GMOs. They multiply and start churning out whatever you've coded them to make, and finally, you harvest that finished product.
Now, I know this can all sound pretty abstract and science-y, but this technology is already all around us. I mean, it's impacting our lives in some truly incredible ways. Let's look at a few examples, from things that save lives to something as simple as doing your laundry.
Okay, probably the most famous success story of all time for biotech is insulin. The old way to get it was by harvesting it from the pancreases of pigs and cows. It was messy, the supply was really limited, and some people actually had allergic reactions to it. But now, with the biotech way, we can take the human gene for insulin, pop it into bacteria, and let them become tiny factories that produce pure human insulin for us.
And the impact of this one change, it's just impossible to overstate. It completely changed modern medicine. We now have a limitless, safe, and reliable supply of this life-saving drug for millions of people with diabetes all over the world. It's the perfect example of how programming a tiny little microbe can solve a massive human health crisis.
But it definitely doesn't stop there. This exact same basic process is used to make all sorts of things. We're talking antibiotics and vaccines, biofuels for renewable energy. We even engineer microbes to literally eat pollutants to help clean up wastewater. And yeah, even in your laundry detergent, those special enzymes that get the tough stains out, they're made using biotechnology too.
Now look, with any technology this powerful, there are always going to be some really important conversations we need to have. And this is especially true when we start talking about genetically modified organisms in our food. So it's fair to ask, what are the trade-offs here?
And honestly, the debate has really valid points on both sides. On the one hand, proponents will tell you that GMO crops can increase how much food we can grow, they can be fortified with extra vitamins, and they can even resist pests, which means using fewer pesticides. But on the other hand, there are some very real concerns about things like potential cross-pollination with wild plants, farmers becoming economically dependent on big seed companies, and the risk of reducing overall biodiversity. It's a really complex issue, for sure, with no easy answers.
All right, so let's do a super quick recap of the key takeaways. Number 1, bacteria are perfect biological hardware. 2, genetic engineering is the process. It's all about cutting plasmids and inserting new genes. 3, we control these genes with genetic circuits using on-off switches called promoters and terminators. And 4, to make it useful, we scale everything up to an industrial level using bioreactors.
So as we wrap this up, the big idea to walk away with is this. We've made a huge shift from just observing nature to actively partnering with it. By treating biology as something we can actually program, we are opening up a whole new world of possibilities.
You know, when you think about everything from medicine and energy to new materials, this ability to design and control a genetic circuit, it isn't just another scientific breakthrough. It really is the engine of the next industrial revolution. The only real question left is, what part of our world are we going to program next?
Goodbye!